Physiology of muscle tone, part 2. To start with, we'll just revise the functional anatomy of spinal cord. This is the muscle spindle present inside the skeletal muscle, which consists of intraphyseal fibers. This is the sensory nerve, which is carrying impulse from the muscle spindle to the spinal cord. This is called as the alpha motor neuron, which is supplying the extraphyseal fibers in the muscle. This is the gamma motor neuron, which is supplying intraphyseal fibers. Now we are giving a stimulus to the muscle in the form of a just tap on the tendon. Muscle will get stretched. Impulse generated at the muscle spindle. It will travel from the sensory nerve to the spinal cord and will excite the muscle through alpha motor neuron. This is called as reflex action. Now if the descending tracts are stimulating gamma motor neuron, now they will stimulate the muscle spindle, leading to generation of action potential in the spindle. Impulse will again travel back from sensory nerve again through alpha motor neuron, leading to muscle contraction. That is, stimulation of gamma motor neuron will lead to muscle contraction indirectly through alpha motor neurons. And if descending tract is directly exciting alpha motor neuron, this will lead to directly contraction of muscle. That's why alpha motor neuron is also called as spinal common pathway. Now we'll just again revise our neurons. Descending tracts. This is a basal ganglion. This is our tract which is coming from cortex ending in the spinal cord, corticospinal tract. This tract arising from the vestibular nuclei called as vestibulospinal tract, which is having a connection with the vestibular apparatus, as well as vestibulocerebellum. Now this tract which is arising from the pons reticular formation is called as the pontine reticulospinal tract, and this tract which is arising from the medullary segment of reticulum is called as a medullary reticulospinal tract. If we just recall, pontine is having a stimulation coming from the spinothalamic tract. It is excitatory in nature, and medullary is inhibitory in nature. Now it receives input from the cortex, this blue line, which will excite it, plus it will receive input from the basal ganglion, which is again excitatory, as well as from the cerebellum. So three top order are stimulating medullary reticulospinal tract, cortex, basal ganglion and cerebellum, and in turn it is inhibitory in nature. So it is inhibitory, it is excitatory. So ultimately they are controlling the muscle tone through this. This is the red nucleus, our rubrospinal tract, which is very similar to corticospinal tract. Now we'll do a brief small experiment in the animal. Commonly it is done in monkeys or cats. We are simply removing the cortex. This is called as removing the cortex. It is called as the decortication. It is called as the decortication. We are removing the cortex now. Now see what is the effect of this now. So when we have removed the cortex now, we are removing one of the influence which is stimulating medullary reticulospinal tract. So this influence excitation goes off. So inhibition also goes off down here. That means one negativity has reduced. So the balance will go more in favor of excitation. So this will lead to rigidity. It is called as the decorticate rigidity. Now what are the features of this rigidity in the decortication? No spinal shock. As the total spinal cord is intact, we will not get typical features of spinal shock once we are damaging spinal cord. So here spinal cord is totally intact, so no spinal shock. Mild rigidity is there. hypothalamic functions are normal because hypothalamus is here. So all the functions of hypothalamus, for example, temperature regulation, thirst, appetite, this all will be normal. But main symptom will be inability to react in terms of past experience, means repetition or recent memory is affected. So these are the classical features of decorticate rigidity. Now we'll go further down in this other experiment. Now we'll totally remove the cerebrum. Okay, we are taking section at the in between the colliculus. It is called as the mid-collicular decerebration. Now let's see what happens in this experiment. So here where we are exactly cutting, we are taking cut at this level. Okay, so now you can see influence coming from the cortex is no more now. Cortex will not excite, as well as influence coming from basal ganglion is also affected. So two positive headers to the reticular formation has gone now. So this feed forward inhibition is decreased now, but still cerebellum is intact, vestibular apparatus is intact. Okay, so mainly basically we are affecting the reticulospinal tract. In that basically we are affecting basically the medullary reticulospinal tract because it is getting stimulation from the up. So see what happens now. So this is called as a mid-collicular decerebration. See here one positivity is gone, so negativity is gone here. Okay, so three negatives were there, now it is only one. So balance more in favor of excitation still. So what happens now? Again there will be increased rigidity. So it is called as decerebrate rigidity, which is definitely more as compared to decortication. In decortication it is mild because only one influence was gone, that is the cortex. Basic ganglion was intact now, but in this decerebration, cortex as well as basic ganglion both, which was stimulating medulla, are now gone. So that's why inhibition is less, more excitation. So this is decerebrate rigidity. Now in this we'll do one more experiment. This reflex arc is there. Okay, now we'll try to remove the reflex arc. So we are cutting this afferent nerve. So it is called as a deafferentation, and we'll see whether rigidity remains or it is lost. So we are cutting this. So what happens now? This is gone. So if rigidity is lost in this, that will indicate that the tracts were influencing gamma motor neuron, because gamma motor neuron utilizes this reflex arc. Alpha motor neuron is directly acting, it is not using this sensory pathway. So let's see, deafferentation, rigidity is lost. Now from this experiment we can have some conclusion that the medullary reticulospinal tract or reticulospinal tract are ending dominantly on gamma motor neuron, because when we have removed the cerebrum there was a rigidity, but when we have removed the afferent nerve, rigidity is lost. That means it was utilizing the sensory pathway to excite the muscle. So that's why after deafferentation the rigidity is lost. So that means reticulospinal tract are dominantly ending on gamma motor neuron. So this is mid-collicular decerebration. No spinal shock as the spinal cord is intact in this animal. Rigidity increases as compared to decortication. Loss of all sensation, because you can see here we have removed this total, means thalamus also gone, cortex gone. Thalamus is the great sensory relay station, so when that is removed, so there is no sensation. Hypothalamic functions are also affected because hypothalamus again here is gone now. And same will be there as decortication. Inability to react in terms of past experience, means recent memory is affected. So these are the classical symptoms of mid-collicular decerebration. Plus rigidity released after deafferentation, because the tracts mostly must be ending on gamma motor neuron. Now let's come to the third experiment, where now we will remove this influence of cerebellum also. Now this practically is difficult to cut, so that's why scientists are using concept of ischemia. So they have ligated basilar artery. So when basilar artery is ligated, all blood flow will go off. So this picture will be like this. So it is called as ischemic decerebration. Means through blocking the blood supply, we are creating a decerebration. So in this, you can see the influence of cortex, basal ganglion, cerebellum, all is gone now. So that means this reticular formation will be no more getting influence from the top. Okay, everything is gone now. So you can see all positive influence on this is gone. So rigidity will be very much increased. And now plus as we have removed the cerebellum also, it is going to affect the vestibulospinal tract control also. Because in the previous experiment, cerebellum was intact. That means vestibulocerebellum was having control from it. Now here this is also gone. So now what happens? Disability is maximum. Now let's see in this what happens if you are doing the deafferentation. So mind well, we have removed the influence of cortex, basal ganglion plus additional cerebellum here. So reticular spinal tract will be no more getting any influence from the higher one now. Vestibulospinal tract again control is gone. Now rigidity persists. Now what it indicates? That means some tracts which were ending on gamma motor neuron, they are also not functioning. But rigidity is persisting means some of the tracts are ending on alpha motor neuron directly. And which is the tract now remains? It is the vestibulospinal tract, because its control has gone now. So rigidity persists means vestibulospinal tract is ending on alpha motor neuron, and through previous experiment we came to know that reticular spinal tracts are ending on gamma motor neuron. So this is ischemic decerebration. Rigidity further increases. Hypothalamic function affected as the cerebrum is gone, hypothalamus is gone. Inability to react in terms of past experience. Rigidity persists after deafferentation as cerebellum removed, and cerebellum was influencing vestibulospinal tract. Now vestibulospinal tract must be ending on alpha motor neuron as rigidity still persists. So we conclude rigidity is mild in case of decortication. Decerebration, it is moderate, and decerebration with removal of cerebellum is severe. So here we are finished physiology of muscle tone.